This is Christina Campbell reporting from Microbiome Times, and we are fortunate to be here today with Xavier Duportet. He's the co-founder of Eligo Bioscience, so thank you for being with us.
Well, thank you for having me.
So can you tell us about your unique approach to microbiome modulation?
So today in the microbiome space, you have technologies to remove specific species in the microbiome, or you have technology to add live bacteria to the microbiome. And I think we're a bit in between where we want to leverage what's already there, specific bacterial population in the microbiome, by delivering into this population a genetic payload that will enable to express in native bacterial population some therapeutic proteins of interest. And so we do this by actually using phage-derived particles that are filled with a synthetic DNA payload, a plasmid, that encodes a gene that enables the expression of the therapeutic protein once delivered in the target bacteria.
Mm-hmm. And so what disadvantages or advantages might this have over sort of live microbes that could be used therapeutically?
So I think it really depends on the target indication. For instance, if we want to specifically remove some bacterial strains within a species, I think it's really hard to do with the live probiotics. But with our technology, we've demonstrated that we can, if we deliver a gene to express a Cas nuclease that is targeted towards a specific chromosomal region, specific to the strains that we're targeted, it would lead to the death of this specific bacteria. And I think so this delivery of CRISPR-Cas system is really one of our lead technology from our platform. But if we really want to compare with what can be achieved with the live bacteria, I think this is something that we still don't know, is whether when we are delivering live bacteria, these bacteria will be able to engraft really well and go into niches where that are really important to have to be able to modulate the host's immune system, for instance. And typically in the gut, it's the bacteria that's where you want to really go to express anti-inflammatory molecules or things like that, is on the epithelial cell wall and to have your bacteria interact with the epithelial cells of the host. And so with live probiotic, we don't really know if this is possible, but we believe that with our approach, actually, we can, because we're not delivering a new bacteria that has to engraft, we are delivering into the existing bacteria. And so therefore, if ever we can deliver in this bacteria and have these bacteria express some anti-inflammatory molecules, for instance, then we might have a much better response.
And so what do you see as the future potential of this kind of therapeutic for health?
So I think there are many. So we're not only, with our CRISPR approach, CRISPR-Cas approach, I think just with this, actually, we can target infectious disease, but we can also target a lot of disease where specific strains are the cause, and the strains of bacteria and what they're expressing might be the cause of a disease. So it goes with with cancer. In oncology, for instance, there are more and more relationship between strains and disease that are being elucidated. In autoimmune disease, in obesity, inflammatory disease, there are a lot. And then I think we can address also a wide range of disease when we express locally, not a CRISPR-Cas to kill bacteria, but to express anti-inflammatory molecules, or even small metabolites or enzymes to modulate the how a drug is metabolized in the gut, for instance. So I think the potential is quite big.
And so from a regulatory perspective, what do you think needs to be understood about this kind of therapeutic in order for it to progress in the regulation?
So I think we are a bit, the pioneers in this field, because we're not a live probiotic, we're not a phage, we're a bit in between, and we had actually we have already discussed a lot with the authorities, regulatory agencies, and we had a very positive feedback, and I think actually it's not so different from other therapies like human gene therapy. where a DNA payload is delivered to some cells, except that they're not human cells, but they're bacterial cells. And so I think safety is definitely something that we need to prove, but we're very, very confident that there is no safety problem, in the sense that we're using bacteriophage particles, and we have billions and billions of phage particles already in the gut or in our skin. And then I think it will be about the payload itself and the genes that you want to express. You need to make sure that the proteins expressed by these genes do not have an effect that you don't want to.
Great. Very interesting, and thank you for being with us today.
Thank you.